Hi everybody, welcome. My name is Lidia Molina and I'm the Early Stage Researcher number 14 and I will be part of the FoodTraNet project for the 3 following years. But first, I would like to thank to the selection committee and M4I for bringing me this opportunity. I will do my best. So, who I am? I am an analytical chemist from Barcelona, and although nowadays I like science and research, I have to say that at the beginning I enrolled a bachelor's in criminology because I wanted to understand criminals' mind. But after spending the first semester, I realized that a science degree might fit better with my personal skills. So then I switched and I became a chemist. One of the things that describe me quite better would be my concern about the environment and animals' well-being, and actually I am a vegetarian. Talking about my hobbies, I would say that I love dancing and I do contemporary dance and dancehall. I consider myself an adventurous person because I like to face new challenges and try different activities. My scientific career started when I enrolled my bachelor's in chemistry in the University of Barcelona, and I enjoyed while I was doing my bachelor's thesis in environmental chemistry, and for this reason I decided to continue working in the same institution during the summer as a laboratory technician. And after that, I started my master's in analytical chemistry in the same university, but I participate in a different project about proteomics. After all these years in academia, I wanted to change, to try something different. And then I started working in a petrochemical company in the harbor of Barcelona called AMSPEC, and there I was able to gain some professional expertise, but also to improve some of my skills. After those months in the company, I realized what I really wanted to do, and it was to develop a career in research. So the first step was to do a PhD in chemistry. Let's take a deep insight in the projects I've participated. The first one during my bachelor's thesis. We were interested in studying the organic contaminants present in mussels from Ebro Delta. And specifically, we were focused in studying the different contamination patterns of these two bays located in the Ebro Delta, and we wanted to identify the priority mixtures of contaminants in those bays. We chose mussels because as filter feeder organisms, they can bioaccumulate chemicals and they can also tell us a lot of information of the contamination present in an area. We were using an on-target approach, an HPLC coupled to mass spectrometry as an instrument, as an analytical technique. And in the end of this project, we were able to conclude that the contaminants that were affecting the most the Ebro Delta were pharmaceuticals and pesticides. The other project I participated was during my master thesis, and it was about developing a new analytical strategy based on solid phase extraction, capillary electrophoresis, mass spectrometry to detect glycopeptide biomarkers. The configuration of this technique is shown in the slide, and we were proposing a new solvent for the solid phase extraction based on phenylboronic acid because we thought that it could interact better with the analytes. And during this project, we were studying recombinant human erythropoietin because it's a very important glycoprotein in doping control and also as a biopharmaceutical. In the end, we were able to develop a new analytical tool, more simple, faster, and more sensitive. Now let's move on. And I will talk about why I'm here today and why I applied to FoodTraNet project. And it was due to four reasons. The first one, because I was really interested in the topic. I wanted to expand my knowledge in microplastics, those emerging contaminants. The second one, because I was really sure I wanted to develop a career in research. The third, because moving abroad was one of my dreams. I wanted to learn about different cultures and different researchers. And the fourth, last but not least, because I wanted to learn about imaging mass spectrometry, a new analytical tool for me and also a new challenge. The topic of my project is focused on plastics in seafood, and as you may have noticed, plastics are surrounding us with different shapes and applications, and it's not surprising that plastic particles end up in the ocean. And some of the organisms that are living there, such as filter feeders and small fishes, are not able to distinguish between the plastic particles and the food, so they end up eating them. And those plastic particles can be bioaccumulated in their tissues, and then they are not only posing a risk to the aquatic organisms, but also to human health. For this reason, it's very important to develop new analytical strategies that allow us to study those contaminants. And in this project, we are proposing imaging mass spectrometry, because with this technique, we will be able to identify microplastics, but also to study the distribution and localization of these contaminants in the biological tissues. I already started doing some experiments with fresh mussels, and here I wanted to show the 4 main steps you have to follow in order to study seafood with imaging mass spectrometry. The first step is to cut the fresh samples into a very thin sections, and you have to do that with a cryotom. Here you can see an example of a section I did with fresh mussels, and here is the instrument. After cutting the section, you have to place it in a slide, and then you have to apply a matrix in order to retain the analytes of interest, and then you have to place this slide in the mass spectrometer, so then the analytes can be detected and studied. Here in the next slide, I'm showing the first results I obtained. The first picture corresponds to the optical image, so it's the mussel before it's being analyzed, and the second one corresponds to the mussel once it has been analyzed with imaging mass spectrometry. Each of the colors represent different regions with different composition of the lipids of the study, and then each of the regions have a different mass spectrum, as you can see in the slide. The following experiments will be focused in studying some polymers as polyethylene glycol and polystyrene, and that's all about the recent experiments. I would like to thank to my research group, M4I, and specifically I would like to thank Ron and Berta, my supervisors, and also Mudita, my colleague. And that's all about my presentation. If you have any questions, feel free to ask. Thank you very much for your attention.